In this video lecture, we're going to go over Chapter 3, looking at the cell. So far, we looked at the chemical molecular level and how atoms can come together by sharing or donating those electrons to form inorganic and organic compounds or molecules. We're going to take all those different structures that we learned already and we're going to start putting them together, starting off here with the cellular level. Now, anytime we look at cells and think about cells, there are actually 4 basic properties or theories that describe cells. And so we give that term that's cell theory. So with cell theories, these are ideas or principles that try to explain something. And so with the cell theory, there are 4 basic concepts that describe cells. The first is that a cell is the basic structural and functional unit of living organisms. And because of that, cells we consider and think of as a small functional unit of life. Number 2, cells will carry out all the chemical activities needed to sustain life. Okay, and that's because our cell is the most basic functional unit of life, and because of that, it will then regulate our basic homeostatic functions. Next is that the activity of cells activity of an organism will depend on the collective activity of its cells. Okay. Now, all these cells will come together, and because they form the basic building block of life, in order for us to grow and function, these cells have to come together. Continuity of life has a cellular basis is our 4th basic concept. And cells, in order for us to have a cellular basis, these cells need to be able to replenish themselves and regenerate themselves. And because of that, cells are really a product of cell division. Okay, somatic cells. These are your digestive, endocrine, all the cells within your body. These are going to undergo mitosis. And the exception to these somatic cells are your sex cells. So that ovum and that sperm that we saw. And these undergo meiosis. Okay. Now, if we think about a cell, if I say the term cell, which you probably imagine is a circular or spherical structure, or also an oblong structure. Okay, so we'll have the general nucleus, it'll have a cell membrane, and all of these internal regions composed of cytosol and these large structures called organelles. However, cells actually are not the same. They all come in different structure, sizes, because they all have different functions for them. Here we have a smooth muscle cell. If you notice, it's nice and elongated. This is actually called a fusiform structure. Blood cells, they can either be spherical if they're white blood cells, or they have this flat and nice biconcave disc if they're red blood cells. Neurons, they have nice long extensions called axons, send messages out to the next neuron, and they can receive communication, receive signals from other neurons called dendrites, so these are nice long dendrites. Cells lining the intestinal tract, they're going to be more rectangular. They have all these cilia on the top with the main function and purpose of absorption of your nutrients. Fat cells, here you'll notice our nucleus is more lateral, and that's because most of our fat cell is composed of all those triglycerides, so storage of all those high energy fats. Now, ovum and sperm, these ones are those products of meiosis. But of course, they're still cells. And our ovum, much larger compared to the male sperm. And here, if you notice, the sperm has this elongated structure here. This is the tail. And this is the only type of structure. It's a whip-like structure called the flagella. And the only one that we see in the human body was going to be from the sperm. Now, as I mentioned, all these cells look different. However, these cells do have three basic structures in some sort of form. And that's going to be the nucleus, it's going to be the plasma membrane, and anything between the plasma membrane and the nucleus and outside of the nucleus is going to be the cytoplasm. And so our nucleus, this is important because it contains our genetic information in the form of DNA. It contains DNA, our genetic information. Cytoplasm, once again, it is located outside the nucleus and inside the plasma membrane. Contains two components. We have the cytosol. This is the liquid portion, and so it's a lot of the water. A lot of our ions are going to be there. Proteins are going to be bathing there. And then we have our organelles, which function as a metabolic machinery of our cell. And carry out all of the functions to make our cell work. Okay, so here's one in green, the Golgi apparatus. Mitochondria, our energy source. Here we have rough endoplasmic reticulum, and we'll get into the functions of all of these in the next video lecture. And then finally, outermost layer here, we have the plasma membrane, and it contains the cell contents and separates them from the surrounding environment. Great. And now for the rest of this lecture, we're going to break down the different regions of the cell, starting off with the plasma membrane. And so that outermost layer, we give it two terms. We can call it both the cell membrane or the plasma membrane. And this forms a barrier, and that does that by creating a bilayer of phospholipids that form a barrier between the extracellular and intracellular space. Okay, so I'm going to draw my cell again. Here's my nucleus, my cell membrane. Anything outside of the cell, this is called that extracellular space. Anything outside. Anything inside of the cell, this is called the intracellular space. Intra meaning within. Another term that you'll often see is if you're looking at two adjacent cells. So here's cell 1, cell 2, cell 3, cell 4, and they can form a sheath-like structure, and you can separate the outside from the inside. This this fluid surrounding all of these cells in this tissue, this is called interstitial fluid. It's a fluid between all of these cells. Okay, so a couple of terminology you'll see throughout the rest of the semester. Now, regulation. What does it regulate? So cell membrane, just like I put this illustration of your house, because if you think about your house, you're not going to let random people off the street into your house, right? Same thing you think about your windows. Not all the windows in your house are going to be the same size. If you think about the windows in your upstairs bedroom, if you have an upstairs bedroom, it's going to be much smaller than the windows separating your backyard or even your front door. And because of that, certain things can enter in and out. So let's say in the very top, you can have some birds come in. Let's draw a bird. Ooh, birdie. Okay. Or those mosquitoes. Okay, great. Mosquitoes, right? They can still fit through those small nets sometimes. If you think about also your garage, if you have a big, let's imagine this is a big garage door for your car. Your car can fit into that garage door, but it can't fit into your front door entrance. And because all of these windows have different sizes, so as your garage door has different sizes, that's how you can think about your cell membrane as well. Certain things can come in and certain things cannot come in. And because of that, the cell membrane works just like your house in terms of regulation. So it can regulate the ions, water, different proteins based on their size. So we mentioned, you can imagine the car being one type of protein, the bird being another type of protein, and a person being another type of protein. All those can go in and out. Some of these openings and some of them cannot. Okay, so ions, water, different proteins in and out of the cell. Structural support. Okay, so structural support, your house, you have bricks, you have wood, lots of wires and other beams getting in and out of your house for that nice support. In our cell, our structural support is going to be that phospholipid bilayer. And cholesterol. Okay, now I realize I didn't go over what this phospholipid bilayer is. Okay, cholesterol, we know it's going to be that steroid, those 4 interlocking rings, nice structural support. And this cholesterol, main function of that is that it gives your cells permeability and fluidity over a wide range of temperatures. So fluidity and membrane permeability. Okay. Phospholipid bilayer, I'm just going to clear our space so we can see better. Okay, phospholipid bilayer. Here is our phosphate head, and we have those 2 fatty acid chains. Phosphate head is composed of phosphate, which is negatively charged, so we have a negative charge there. Our fatty acid chains are composed mostly of hydrocarbons, meaning we have carbon, 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 hydrogens at the ends of all these carbons. And because this is no charge at all, it doesn't want to interact with charged species. Meaning it doesn't want to interact with water. And because of that, so this is, if it doesn't want to interact with water, we give that the term hydrophobic. And so these fatty acids form this hydrophobic tail. And because our phosphate is charged, water has a partial negative and a partial positive side. And phosphate is going to interact with all these partial positive hydrogens through that hydrogen bond. So it's not a direct connection, it's just that nice attraction. And eventually what you'll see is this membrane surrounding our cells are going to be composed of all of these phosphate heads being closer to water, since they are charged and they like being close to water. And of course, since we have this hydrophobic tail, we don't want another water in this area. So what are we going to do? Create this other layer of phospholipids, all their tails facing towards the inside. And this is that phospholipid bilayer. So here is phospholipids, layer number 1, and layer number 2. Okay. Now, if we look at this phospholipid bilayer, you'll notice also we have all these proteins that are integrated within our layer. Okay, and so all of these proteins, they're not stagnant. They can move. So this protein can stay here, or it can move down over here over time. These ones can move also from another area to another area. And because this membrane is continuously being regenerated, all these phospholipids, and these proteins are moving, we give this term, or we call this plasma membrane a mosaic. We look at it as a fluid mosaic model, and that's because this is continuously being moved around. And if you've ever seen a mosaic, it's nice and pretty. We have lots of different colors, lots of different pictures, illustrations going on, and it looks kind of like a hot mess, right? And so just like a hot mess, that's the same thing as our plasma membrane. If you look at it from an aerial view, looking down, eyeballs go this way. Here's your eyeball looking down. You're going to see that it also creates this nice, pretty image. You have proteins, you have other things that we have here on top. We have glycolipids, glycoproteins. Basically, that means you have your protein that can be integrated within your plasma membrane, or it can be a surface protein like this one over here. And you have all of these glucose chains attached to them on the surface. You can also have glycolipids, meaning this is that sugar groups attached directly to your phospholipids. Then here is that cholesterol, and cholesterol needed for that membrane fluidity. Alright, and so let's look at filling this in. Two regions. We have the polar head, being this layer over here. These are all the polar heads. And we have hydrophilic tails, being these little tails that you see all in white. Molecular components, we have lipids, proteins, carbohydrates, and they function, specifically these proteins, they have different roles, such as enzymes, receptors for hormones, their binding sites for anchoring cells to internal fibers. And these glycoproteins can also determine your blood type. So lots of different functions. Okay, so we looked at these cell membranes, the structure, and how do we then join one cell to the other? That's another role of the cell membrane. So we can do that in 3 different ways. Let's go back here. All these glycoproteins. Glucose in itself, you think of something as sugary, it's actually very sticky, and because of that, it can act kind of like a glue. And so that's one way that we can help bind the 2 cells together. So those glycoproteins act like a glue to adhere cells together. Another way is just looking at the contour of the cell itself. So just like 2 puzzle pieces, how they fit together, cells can also do the same. So wavy contours of membranes of adjacent cells fit together like a tongue and groove fashion. So here's a tongue, here's the groove. And then finally we have what are called membrane junctions. And this is the most diverse category. And so the best example I saw for this is if anytime you look at a car seat, you'll notice there's a lot of stitching. So we can bind together one sheet to another sheet, and if you imagine this is one cell and another cell, you can either sew it on together, you can glue it on together. If you imagine this hole going through all to the other side, these holes can connect and create this nice pore between the 2 different sheets, and that's how cells can work as well. The first type of membrane junction we have are tight or impermeable junctions. And the function of this is to prevent movement of substances between cells. Okay. Now here we can sew. I gave the example of kind of like a sewing machine or someone hand stitching two things together. And so here if you have one cell, you have your other cell, you're pretty much stitching and sewing together these two cells together. Now a stronger way to connect these cells together are by desmosomes. And these are called anchoring junctions. And they help withstand mechanical stress. And so right now I want you to pinch your skin and try pulling it apart, right? So you're probably not doing it, but just try. Okay, there you go. Okay, so pinch your skin and try to pulling it. And now if you notice, the cells in your skin are not going to start falling apart. And that's because of all of these desmosomes. And so here's one example of when people undergo construction of a building. You have these nice long rods that are mounted from one area to the other, and you fill it up with concrete, and this gives it nice mechanical strength. Same thing between these two cells. If here's one rod, it's going to enter and go out the other way. And so here you have this nice connection between the two cells, holding that in place. The third type of connection that we have between membranes are going to be gap junctions. And really this is not a structural support as much as it is a communication way, a means of communication between two different cells. So it's means of communication between cells. Okay, and so this entire gap is called a gap junction, but it's formed by proteins called connexons. Here's an example. And so if you want to connect one side of a mountain to the other, if you don't want to jump over it, well, what can you do? You can go through it. And so here is the connexon, which is the protein forming the tunnel. And then the gap junction is just the entire structure itself, including this tunnel and canal in which your cars or different things can go from one side to the other. And so with this gap junctions, what can we transport and transfer from cell to cell? We can transport nutrients and ions. Okay, so 3 different things. All right. Now, this idea that the cell membrane is just like a house. Oops, okay, let me just draw my house instead of my cell membrane. Having the garage, having the windows, having the front door, and allowing things to go in and out, that underlies this idea of selective permeability, meaning that the cell membrane will allow some materials to pass while excluding others. And it will move nutrients into the cells, but will keep undesirable substances and wastes out. Okay, so we go to Costco all the time, right? You want to stack up, load up, and all that good stuff. So you always bring food in, you bring all these nice things to like your toilet paper, your paper towels, all those things needed for your everyday uses. And of course, if you're done with your waste, you flush the toilet or you take your garbage out. So same thing in the house, just like that cell. Permeability factors. Okay, so not everything can come in. And what are some permeability factors? We have size. So only the car can go through the garage, not the window, the front door, the front window. So we have molecular size, if it's small or big. Another thing are electrical charges. If it's charged or uncharged. Finally, if it's lipid or water soluble, meaning can it dissolve in lipids or can it dissolve in water? Okay, and so anytime you see that phospholipid bilayer, because this middle part is composed of lipid, what is soluble, what will enter into the cell very easily are lipid soluble molecules. Proteins, charged species will typically need to be carried in. Okay, and then finally, things can come in if it's assisted by some sort of protein membrane carrier. Okay. So there's different ways we can transport things in and out. It can either undergo through its concentration, so meaning that just from a higher concentration area to a lower concentration, and without the use of energy, and this is going to be passive transport. Now, if we need to bring something in or out of the cell and it requires energy, that's going to be active transport. So passive, think about just chilling, no energy needed. So it does not require energy. And if you're actively doing something, it's such an effort, so it will require energy. There are two different types of passive transport we're going to cover: diffusion, lots of different types of diffusion, and then filtration. Okay, so this first concept, this is the most basic concept of how things can enter in and out of the cell if they are able to pass in and out of the cell, and that's just by simple diffusion. So this is random movement down a concentration gradient. What do I mean by that? Okay, so here, if you put a concentrated block of purple dye into a beaker of just normal room temperature cold water, over time what you're going to see is this block, since it's highly concentrated with purple dye, eventually it's going to go from a higher concentration state to a lower concentration state. And it's really this movement of water molecules moving it back and forth, bumping these purple particles together, and so since it doesn't like being together, eventually it's going to start separating. So if you think about being in a room full of, like, I don't know, 100 people, and the room is huge, would you rather be next side by side to somebody and feel their body heat? No, over time you're going to want to spread yourself out so you have your nice personal space. Same thing with these water molecules and all of this dye. Over time, this high concentration, this is you all being pushed into one corner. You're going to want to spread across the room and have your nice space. Okay, so in the end we have this nice homogeneous mixture, meaning this equal, uniform mixture within the entire container. Okay, so particles, all of these dye particles will tend to distribute themselves easily within a solution because of kinetic energy. So kinetic energy just deals with the movement, energy of movement. And to kind of give you some background about that, here you have your water molecule, partial negative, partial positive. Because water molecules will consistently act with each other, remember we're going to have partial negative, partial positive, partial positive. We're going to have transient, very short, temporary hydrogen bonds, and they're going to continuously move and align themselves. So as this is moving and realigning itself, that in turn is going to help push all these particles in different directions. All right. Next thing, looking at diffusion through this plasma membrane. The first idea is simple diffusion. And this is just movement from high concentration to low concentration. And so here we have the membrane. If you have more molecules on one side, over time it's going to go to an area where there's not a lot of molecules. So in time it's going to start going towards this direction. If we start off with 8 molecules of lipid in the very beginning, all on one side, we don't have any in the inside, what's going to happen over time? Well, we're going to have some that are going to start transversing our cell membrane and enter into our cell. And of course, this is going to go back and forth, so maybe this is going to have 6, and eventually it's going to go back here. And so finally we have equal portions where we have 4 and 4, right? Homeostasis, nice equal levels. Another type of plasma membrane diffusion we have is carrier-mediated. Facilitated diffusion. Carrier-mediated, meaning that in order for these molecules to enter into our cell, they need to be carried in by some sort of protein carrier. Okay, and so lipid insoluble, and remember I was mentioning lipid, anything that's lipid easily crosses through a membrane. If it's not a lipid, it needs to be carried in or it needs to be transported in somehow. So here, this does not require any energy, but we do need to bring things in and out of the cell. Here's an example of glucose, 1, 2, 3, 4, 5, 6, 7, 8. 8 molecules all in all. Over time, we're going to start bringing this in until we have 4 molecules here and 4 molecules on the inside. Now here we have channel-mediated facilitated diffusion. And of course, it once again is lipid insoluble, can't cross. But this one, it doesn't need to be carried in. So it's small enough just to go through this little canal created by our protein. And it's usually by your size and your charge. And so ions, you have sodium channels, we have potassium channels, calcium channels. They can easily allow these ions to go from the outside towards the inside. Okay, so what's really important take-home message once again is both types of these facilitated diffusion in both of these types, the substances are lipid insoluble and too large to pass through the membrane, or if they are charged too, charged species. Okay, so 1, 2, and this third one we're going to go over is diffusion of water, and this is called osmosis. Okay, so with these two, we see the movement of molecules or solutes. Here we're only looking at the movement of water. Okay, so osmosis is the diffusion of water through a specific channel protein. This is called an aquaporin. Here's an aquaporin. Or through the lipid bilayer. So we can either go through this carrier or it can immediately just cross through this lipid. Now here with water molecules, let me just give you an example. Let's say that we don't even look at these other protein carriers or protein channels, and we have 4 molecules of glucose outside of our cell. Inside a cell, we have 0 molecules of glucose. Okay. Water will always go to where there's a higher concentration of solutes or molecules. And here outside, because we have 4 molecules, what's water going to do? Water is going to come from the inside of the cell and go towards the outside. So at the end of this, we're going to have equal concentration overall of your water and molecule ratio. Okay, so It's all going to go outside. And we'll get into hypertonic, hypotonic, and isotonic solutions, and that's really this movement of water and how that can affect the shape of your cell. Okay, so here is more clarification of this idea of osmosis. Osmosis, this is only movement of water, once again, through a semi-permeable membrane. And so this is really important, is that this membrane can change within the pore size, or it can be large, or it can be small. But basically what that means is it allows some solutes through and not some other solutes. And so here, let's look at glucose molecules. We have more glucose molecules on side B compared to side A. This membrane does not allow glucose to cross. That's why it's selectively permeable to glucose. And the only thing that can cross, that's small enough to cross through this membrane, are these water molecules that you see in blue. So over time, what do you see? Well, water is going to start going towards the area where there's more glucose concentration. So at the end, what's going to happen is we're going to have equal ratio of this water and glucose on side A and side B. This over here, all this is showing is that you can prevent, all this is saying here is that you can prevent all this water going on this side by applying some sort of force and pressure. But under normal circumstances, all this water is going to start going towards this direction unless you somehow stop it. So you stop it, apply pressure, you're going to want to have all this water go this way, but it's going to get stuck. It's not going to be able to enter into this direction. Okay, so that's the idea of osmosis. Now the next type of passive membrane transport is going to be filtration. And filtration is done as water and solutes are forced through a membrane by fluid or hydrostatic pressure. Okay, here this great example is within our kidneys. Here you're going to have blood going into your kidneys. It's going to get forced through, you have this smaller area, smaller vessels. Anything small enough to fit through these vessels are going to get forced out, and it's all because of your blood pressure. Blood pressure is going to start forcing out all that liquid portion of your blood into our kidneys structures. This is called a nephron. And then everything, we're going to start returning that blood back towards our heart. Okay, so it's only the movement of this pressure generated from our heart that's going to force out all of this fluid. Another example you can think of is if you get a beaker, you put a paper towel in here, and if you want to strain anything, so let's say you made some lemonade, right? You put some lemonade, you put this paper here, you have some seeds, or you don't want any of that pulp, you only want the liquid, you can pour down your liquid on this paper towel, and the only thing that'll get through is a liquid portion. You'll collect the pulp, you'll collect the seeds. So really this is also an idea of filtration. It's this force, this atmospheric pressure that you've generated, and also the force of pouring that fluid in that's going to cause and drive all this fluid to get through your entire paper towel. So this paper towel is going to serve just like that membrane. It's going to be this barrier to allow certain things in and not other things, larger particles. All right. Now, this next portion is going to require energy, and this is going to be the active transport. And there's two different times that we are going to require energy for filtration. And so it's going to require two things anytime you need energy: a protein carrier and ATP, because we need that energy. Okay, so substances that are unable to pass through by diffusion are going to be needing energy. Also, the most time you're going to see this are when substances move against a concentration gradient. All right, let's get some examples in here. Okay, so this is one example called the sodium-potassium pump example. Under normal conditions, we're going to have, this is outside of the cell, and so outside of our cell we have a high amount of sodium, so high concentration of sodium. These brackets just mean concentration. Inside of our cell we have a high concentration of potassium. Okay. And why do we need this? Well, we'll get into that for our muscular and nervous system. Okay, so here we have high sodium outside, high potassium inside. Looking at sodium first, where does sodium want to come in? So we have high sodium outside, it wants to go into the cell, and that's just by simple diffusion. Potassium inside, we have high potassium. Where does it want to go? It wants to go outside of the cell. Okay, this is under simple diffusion properties. Now, with this, because this is called active transport, that means that we're going to go against the gradient. So sodium's going to want to come in, but we really want to keep sodium out. So how do we do that? Once those sodium molecules come in, what can we do? We need this protein carrier. We can attach those sodium molecules in that protein carrier. We give it an energy source in the form of ATP. ATP is going to break off one of that phosphate. It's going to give off energy and ADP as a product. It's going to cause this to shift. Now we can put sodium outside of our cell. Sodium, potassium. So now we're looking at potassium. Potassium is going to come into our cell, and then it's going to enter. Okay, and so this is how we can maintain potassium inside and maintain sodium outside. And once again, it's against its normal simple diffusion or normal concentration gradient, which is why it requires energy. The second time we're going to need energy is when we use a vesicular transport. Okay, and so in this instance, sometimes if the cell wants to take in fluid from the outside, it can actually cause part of its cell membrane to come in and pinch to form a vesicle. So vesicles are membranous sacs formed from the plasma membrane. In this example, I went over pinocytosis, so this is what's considered cell drinking. And that is, you can bring fluid in, or you can also bring in proteins and fats. Okay. Other things too, anytime you have a bacterial infection or if there's something else, some debris, you can also bring that in. And if here are our proteins, and so of course, if you look at your proteins, your protein carriers, here's your bacteria, that's not going to be able to cross through your protein carriers. So in order to get that into our cell to break it down, we need to engulf it. So here we can engulf, if this is a bacteria cell, we can engulf it, bring it into our cells, and eventually break it down. So this is called phagocytosis or cell eating. Okay, that's when you do large particles, such as bacteria or dead body cells. Okay, so if the cells have died or if it has exploded or anything, you can take up some of that debris into the cell. Okay, so this is another way that we can move substances in or out of a cell via a vesicle. Okay, so we can either allow these large components to either come in just by coming in this direction, and the membrane can pinch it off, or these substances can bind to receptors, and by doing that it'll initiate a chain of reactions to bring that cell within, bring this substance within the cell. And so here we can give this term a ligand, which is just like that substrate. Ligands will bind to receptors, anything that binds to the receptor. Receptors are specific to these ligands. It's going to form this vesicle through that process of endocytosis. So endo meaning within, so bringing it within, and cytosis engulfing that. Here we have this vesicle that has been formed. And if we're going to break it down, we can fuse it with something called a lysosome. It's one type of organelle, and this contains digestive enzymes. So especially if this is a bacterial cell, you can bring it in, fuse it with your lysosome, and over time it's going to start breaking it down. Okay, and then you can expel it into the extracellular environment, or it can just remain within the cell. And this process of after fusing that vesicle with your lysosome, you can then bring that vesicle back towards your extracellular environment, release the contents, and this vesicle itself can then become part of the cell membrane again. Okay, and this is a process of exocytosis. So exo, exiting, removing that vesicle, allowing it to fuse within this membrane. This process of exocytosis uses what are called snares. Okay, so snares, I took this illustration of one of those wires that you find on your bread. But if here's one of those wires found within your membrane and another one found here within your vesicle, V snare, this is the one found on your vesicle. T snares, these are ones found on your cell membrane. And all it's going to do is going to start coiling up just like this and expel the contents to the extracellular environment. Okay, so that's how exocytosis works, using these snares. Here's an electron micrograph of this vesicle fusing with the cell membrane and releasing the contents through exocytosis. Now the second region that we're going to look at is called the cytoplasm, and the material this is all the material once again outside of the nucleus, inside the cell membrane, containing three major elements. Earlier we mentioned cytosol and organelles, but in the event that it is a fat cell, we have another one called inclusion. So cytosol, we had organelles, and we have inclusions. Okay. So cytosol, it's a fluid that suspends other elements. It contains the nutrients, metabolites, ions, and proteins. Organelles, these are the metabolic machinery of the cell. And just like our body, we have organs. These are all considered the little organs of the cell. And then inclusions, these will store nutrients or cell products. Okay. So lipid droplets, glycogen granules. All right, and so the different organelles that we have we're going to go over next class. And if you want to watch more videos on osmosis, passive transport, filtration, the sodium-potassium pump, and understanding endocytosis and exocytosis, please click on the links in the bottom of the video lecture, and that'll help you understand those concepts.